Okay, final video for part one, hopefully. We talked about experiments versus observational studies in class. This is another thing, possible test question, identifying whether or not something is an experiment or an observational study. Again, not terribly hard to do. Just keep in mind that in an experiment, we're going to impose some sort of treatment. So if we are assigning people to do something, or to watch something, or to read something, or whatever it might be, then we're doing an experiment. If we're just allowing people to self-select whether they are going to take that or read that or watch that, that's an observational study. And that's going to have some of that selection bias we saw or talked about with the CT study. Okay, so I'm going to give you a moment. Pause the video. Try the first exercise. All right, so hopefully this is an old exam question. This was easy. Sometimes students have a mistake here. We have a group of adults who swim regularly that are evaluated for depression. They seem to be less likely to be depressed than the general population. In a second study, a group of 100 volunteers was randomly divided and then asked to swim. So in the second study, we are asking people to do something, which makes this an experiment. Well, in the first group, we just found people who swam regularly already, so we observed them. So B should have been your correct answer, observational for the first, experiment for the second. If you chose A, likely you just got distracted by our treatment group and our control group within the second study. So just read carefully. The second study is the whole second part of that. Don't get confused by the treatment and control groups. Observational studies fall into three different categories. Cross-sectional, all your data is collected at a single point. So you just go and ask or do things all at once. Retrospective study asks about the past. So usually these involve finding a group that do whatever it is that you're interested in or have the outcome that you're interested in and go backwards to see what influencer might have, been a part of why they're in that group, why they're different than the other group that doesn't. Retrospective studies, we'll get in here. Retrospective studies are very useful because you can use them for rare diseases and things that don't occur very often. You can imagine if you're doing a cross-sectional study and you want to investigate, something that is very, very uncommon. Say you take 1,000 people in a cross-sectional study, which is a pretty big sample size. If it happens in less than 1% of the population, you may only get a handful of people who have whatever outcome you're interested in a cross-sectional study. So instead, you would find the people who have that outcome and then ask them some questions and get a similar healthy group and ask them questions. Prospective studies, you follow people forward in time. This is similar to the longitudinal study that I was a part of at UCSD. So usually we follow them and we take at multiple time points some information, or we just ask them to keep going until some outcome occurs. So you have an example here about drinking alcohol and whether or not it increases the risk of coronary heart disease. So they followed them from 1993 to 2002 to see if anything like that happened. So our exercise here, I love to ask, which one makes the most sense for a particular situation? So if you're studying factors that might impact a person developing brain cancer, which of the methods would you use and why? Brain cancer is not very common. So to me, this leads straight to a retrospective study. You could get a sample and then follow them into the future in a prospective study, but you'd be following them for a very long time. And there's no guarantee you'd get a large enough sample size to be able to do anything meaningful. And a cross-sectional study, you're going to be in the same boat, except for you don't have to waste all the time waiting. So retrospective study would be the best one here. Oops, rare. So I'm going to use the bottom half of this and just sort of do a quick little retro versus prospective and just kind of throw down some positives and negatives to each, pros and cons to each. For your retrospective study, big positive is that we can look at rare outcomes. It's really the only way that we can look at certain outcomes. The negative, though, is since we're asking about the past, we're going to have a lot of recall bias, right? We even saw with that little research thing we did on the first day. We couldn't remember what time we woke up on Sunday, right? I had no idea what my answer to that question was. I had a rough guess, but there's going to be a lot of recall bias if I'm asking people about things that they may have done in the past or how often they did them or when they occurred. With the prospective study, the biggest positive, since we're following them through in time, we have lots of positives. One is we're going to have really good data, right? If we're following them through in time, we can ask them to keep a journal. We can ask them to come, like my VETSA group at UCSD, we can ask them to come in every 5 years. So good, clean, happy data. Additionally, we're going to have the time component, right? So we may be able to even investigate how things are progressing and do some interesting analysis that involves that time and time to an outcome or something like that. So that time component is useful and interesting as well. Our negatives for the prospective study, definitely the time and money. I mean, you can imagine being that Denmark study that started back in 1993. You're like, well, this will be great. In 9 years, we'll be able to, write some papers. Sort of like VETSA. Every 5 years, they're doing a new study. So, in 20 years, there'll be 4 time points and they could actually start looking at some of this stuff. But yeah, so the time and money is probably the biggest negative of the prospective study. Additionally, you can't use it for rare outcomes because then you're not going to get any people in that group. So I don't tend to ask many questions about those observational studies on exams. The definitions are pretty straightforward. But I do ask sometimes about the types of experimental design. So our experiments, when we impose a treatment, we have to randomly assign people into those treatment groups. Otherwise, we're going to get in trouble in terms of, not having results that we can make conclusions from. So that random assignment is got a lot of similarities to our sampling designs. So there's experimental designs. There are many, many more than this, just like there are more sampling designs than what we saw. But here's 3. Completely randomized just means we take our whole group of subjects and we just randomly assign them into the treatment groups and then do our observations. This is probably the most common. Now, it may turn out that we are interested in making sure that there's a similar number of people in each group. This is very similar to stratified, right? If we are worried that it could end up where all of our men end up in this treatment and all of my women end up in the control, or that the numbers are really far off, what we can do is we can take our subjects and then divide them into groups that are similar, just like we would in a stratified. And then from that, then we do our random assignment. Big difference between it and stratified, the random assignment versus random sample, right? So it is important that you distinguish the two. Everybody here, all of our subjects are people that we are going to be getting information from. So we've already done the sample portion, and now we're doing assignment versus in the stratified. So once we do a random assignment, then we go ahead and assign. Then we go ahead and do our measurements. The last here is a matched pair design. In a matched pair, it's basically a subset of our block design. The big difference is each group only has 2 people. So if I choose one person out of our class and assign them to go to the CT session, anything that I think might influence how that CT session works, anything I want to control for, I am going to go ahead and say, okay, look, this was a Hispanic male, he's 18, and he came in with a GPA of, I have no idea, 2.2. So what I'm going to do is I'm going to look through the rest of the class and try to find somebody else who fits as similarly as possible those parameters. And if I can find somebody, I'm going to take him and put that person in the control. So I'm going to have one guy in the treatment, one guy in the control that are similar for the things that I think that might influence how that CT session works. So similar to the group, but each group, similar blocking, but each block has just 2 people. These can be tested in some less obvious ways. A before and after study is a very subtle example of a matched pair. In a before and after study, you come in, and at the beginning, we have a baseline for you. Then we give you the treatment and get an after. So we actually have a pre and a post. So you act as your own control. You are your own matched pair. Twin studies, a lot of them would end up in this category as well. So those are just two examples of when we might see matched pairs happen. And I deleted a whole page that I want to have in here. What did past me do? All right, so I am going to, I guess, do a separate little thing on that in class. So there we go.